And if I gloss over some technical details and you want me to go back and explain more, please just like interrupt and say, hey, wait, can you go back and describe how you did that? Because I've been giving the presentation to a more general audience. But today I'm hoping to sort of like take a step back and talk about how we developed some new science and a new model, and then what the process was like for us to put that into production and then try to develop it into a more generalizable tool. So in this case, the project will be applied to in the context of wastewater, but I think a lot of this is more generalizable to really how we're integrating novel data streams and having new sources of data, potentially new pathogens and new problems, and how we can basically translate development of science into what we're interested in like this community, which is productionizing pipelines, producing regular outputs, and then developing tools. Okay, so I just wanted to start with introducing the team a little bit. I'm not going to describe everyone's roles in detail, but just to highlight a few, Dylan Morris is the team lead of the short-term forecasting team here at CFA, which is under our Predict division. So he's been sort of overseeing this project since it became a forecasting project. And then Scott developed some of the earlier wastewater science, and he is on the medical countermeasures and variance team, also a team lead. And then Sam has been pretty involved in this project, giving me lots of great feedback and lots of quips, and also really helpful because this is built off of EpiNow2 infrastructure. And then I'll just highlight Christian, who is a summer intern with us at UC Irvine. He's been working on developing spatial components of this model, so I'll talk about that, the sort of extensions off of the initial model at the end. And then just highlighting a few others, Damon, who's been working on a more general framework for doing integration of multiple signals. And then our collaborators at News, so that's Heather and Dan, who have been really helpful with helping us understand the wastewater data and get access to it. And then lastly, just wanted to give a shout out to Kayla and Isaac, who are our Inform colleagues. So they are focused on state and local engagement and sort of getting all this work into like in front of state and local audiences and getting feedback from them about how we can make this more useful to them, how the outputs could be more useful, but also trying to get feedback on how the model can be improved and how they can interact with it. And then I put those two links in the chat just for anyone interested and wants to take a look. We're developing a modeling package that will be intended for those state and local partners who are analyzing wastewater data themselves and wanting to produce their own forecasts, as well as our code for actually producing them ourselves. Okay, so the plan for like this talk is basically to describe sort of the process that we took from having this scientific question of like, wow, wastewater is really cool. We want to use it. How do we do that? Here's one way we can think about it. Okay, how do we now turn that into something that has a practical application? How do we turn that into a pipeline and do something in production and deploy that science into a framework that can produce regular outputs? And then this sort of life cycle that we followed, which was then, okay, how do we make this more generalizable so that it's not just us producing outputs, but we can empower others to modify the model, change it, and then also just run it themselves when they want to. And so I kind of want to frame this discussion around like, how do we in this field sort of generally streamline this process? Where are like the pain points, and just along those lines, how can we do better at this next time? So this is a lot of words, but I just wanted to like start up front with discussion questions. So just sort of like framing what I'll talk about, which is I'm going to kind of describe in a linear fashion how we developed this model and the steps that we took. And then I want sort of like to have a discussion about how we can sort of streamline this process of taking science and moving it into this production and tool development space. So some things that I've been thinking about are really the trade-offs between tools fit for purpose for a very specific problem versus tools that are maximally modular. And can you start by solving a specific problem and then generalizing it, or do you always have to build towards maximally modular tooling? what relatedly, like what are the differences between tools for modelers? So tools intended for people to build on new features that are specific for their needs, and tools for, say, decision makers who are going to be looking at the outputs and using them. And how do we decide who our users are when we're developing these new scientific models? And then in a world where we need to show value early and often, I think one of the things that we learned from this project that I'll describe a bit is that it can be helpful to develop a proof of concept, but how do you know when it's ready? And how do you sort of transition from something that is a prototype to something that we want to do longer term planning and scoping, and kind of make sure that we're following all the best practices and actually turning something into a real product, shall we say. Another thing that I'll touch on is really, a question for the group is like, should model evaluation be built into our development processes in the same way that we build in things like continuous integration and testing and documentation? And if so, like, how do we sort of push that forward? This was a real struggle for us, which I'll discuss. And then as I'm talking, like, I have an idea of what we should have done differently, but it would be great to hear from everyone else. Like, what do you guys think we should have done differently? Okay, so I'll go ahead. I'm going to describe the wastewater data in the US, since it is kind of different everywhere. And then I'll talk about our initial goal, which was to produce these weekly state level COVID hospital admission forecasts, how we developed that model, and then how we're evaluating the performance of that current model. And then lastly, focus on like some of the things that we're trying to extend this model to do, and some of the challenges that we're encountering now. Okay, so just to give an overview, on the left is a screenshot from the CDC's news wastewater dashboard, where each dot on this map represents an individual site, and the colors represent the trend. And so the point here is really just that there's lots of heterogeneity in the number of sites per state, and the proportion of the population that covers. So in some states, there's over 100 sites, and others there's none or one site, and that can represent just a very small population size. But there's also lots of heterogeneity in the populations that are being covered by wastewater surveillance in the US, which provides a lot of, I think, opportunity for us to have sort of these localized data signals. And then on the right, I'm just showing an example of a snapshot of data on the viral concentration in wastewater from the state of Indiana at a certain point in time. And the point here is just to sort of say that in both instances, this data is kind of hard to interpret at face value. This has been a challenge, I think, for a lot of the field in wastewater, where it's hard to see the value in data if you can't look at it and see that it's telling you some type of information very clearly. And I think of this as like analogous to right truncated case data, because it's the same problem where if you look at the data alone, you might not be able to get information because you can't tell. Our brains can't do that delay estimation in our heads to see that, oh, it's actually truncated less than it was previously, therefore trends are actually increasing. And I think this is similar, where there's lots of complexity here. And in order to gain signal from that data on the right, this really is like a perfect use case for our field, which is developing like statistical models with infectious disease modeling frameworks that can sort of do the right type of analytics to make this information more actionable. And I just wanted to like give a little bit of a hype on wastewater, because I think while it's like has a lot of complexities and challenges, it's really exciting and awesome, and a really, really cool new opportunity for new science to be developed on it. So one of the great things about this data stream in the US is it's continuously updated. So there is not a standardized collection frequency or reporting frequency, but this means that every day this data is coming in. It's not, of course, like not biased by healthcare seeking behavior. So this is just like a passive surveillance method, which is great, especially as things like, we all modeled cases during the COVID pandemic. And of course that can be difficult when suddenly like tests are less available or things are changing. And so that's one of the other benefits. It's also relatively low cost. It can provide us with highly localized information. And then the other thing that I think is really important as we're thinking about real-time outbreak tools is this is based on PCR targets. And so it is very easy when this infrastructure, not very easy, but it's relatively easy when the infrastructure is already set up to test for one target to add additional targets. So when we have novel pathogens, we can actually, novel pathogens or existing pathogens where we are starting to see outbreaks, we can actually add in targets. And I think this is really exciting because it means if we can build analytical infrastructure using wastewater data, we'll be way more prepared when there is a novel outbreak to actually get information from this data. So given all that motivation, wastewater is really cool. We wanted to work on it. We were like, let's do some science on it. But also we have to produce something, right? So the first thing that the obvious outcome that seemed the most feasible was, can we use wastewater data and hospital admissions data to jointly infer incident infections and then use that to generate forecasts of hospital admissions and submit them to the COVID forecast hub. So that was our initial goal. We started kind of, I guess it was around a year ago, thinking that, okay, now we want to turn a model into an actual production ready forecasting pipeline to submit to the COVID forecast hub, which accepted submissions every Monday for 28-day ahead forecasts. And each Monday, the NHSN data, which is the mandatory required COVID and flu data set in the US, was available up until the previous Friday. And so we had a 9-day delay between the last hospital admissions data and the forecast date. And so our hypothesis and kind of the motivation behind using wastewater to produce these forecasts was that because those hospital admissions represent infection trends from well in the past, perhaps wastewater could tell us about more recent infection trends, and that would be useful to perform, to produce more accurate forecasts. So that was sort of our goal going into this whole endeavor. So where did we start? We started modeling the wastewater data as just another signal coming from incident infections. So in the same way that in EpiNow2, we model hospital admissions as a signal off of a latent process that is our incident infection time series generated via a renewal process. We do the same thing here where we say that really wastewater is just another signal with a delay distribution and some scaling factor that impacts how we actually observe that signal. And so fecal shedding is just another signal that we can use to jointly infer the incident infections. And so we started with the simplest model where we took an assumed and not realistic data stream where we had an estimate of the viral concentration on average in an entire population, as well as a hospital admissions data stream for that entire population. So in this case, we actually started with regional data from BioBot. BioBot, as of this time last year, was producing estimates of a population weighted average with some thresholding of the viral concentration in a region. And we took that like somewhat manipulated data as our wastewater data stream. And then the hospital admissions from the individual states aggregated across the states in a region to get a hospital admissions data stream for a region. And then we sort of developed the model trying to see how can we jointly infer these two signals coming from a shared and same incident infection time series. So just like EpiNow2, which is like basically this whole top thing, although we did add some modifications to the RT time evolution and also to the like we added an infection feedback term and we changed it to RT has like weekly change points. There's a few modifications, but it's basically like a lot of the same infrastructure as EpiNow2 in this hospital admissions module. And this is the same idea that we did for the wastewater generative process. So we're just assuming that individuals infected shed virus with some shedding kinetics distribution described by S of tau And then that gets involved with your incident infections to generate your total genomes, to generate the delayed, and then you scale by the total genome shed per infection and divide by the volume of wastewater produced per person. And so this is just a deterministic transformation from your incident infections. But like what we actually have, as I described, is not that structure at all. So in reality, we have one signal, which is that state level hospital admissions that's coming from an entire state population. And then we have the populations in each wastewater catchment area that are contributing to the observed site level wastewater concentration. And these represent subsets of that total state population. And so this structure is really a perfect structure for setting up a Bayesian hierarchical model with partial pooling. And so that's what we did. We basically made the assumption that the wastewater concentrations observed in individual sites represent signals coming from localized outbreaks, so likely to be related to one another, but not exactly identical. And so we modeled the site level RTs, or the subpopulations represented by the sites, as deviations from the state level RT. And so from the site level RT, we generate an estimate of the incident infections in each site and also in the population in the state not represented by the wastewater data. And then those incident infections generate expected wastewater concentrations. And then we compare those to the observed concentrations. And then the incident infections in each of the subpopulations are summed up to get the state total incident infections, which then give rise to the state level hospital admissions using that convolution with the delay distribution and the IHR. And so there's sort of two reasons that this structure was something that we were excited about. One is that, of course, we think that the wastewater concentrations should be telling us about infection trends, potentially with an earlier signal from incident infections. And so it could help guide the estimate of the infection trends that generate the state hospital admissions. But additionally, the partial pooling should also constrain the estimates from the site level wastewater concentration. So in an ideal world, this would actually be providing more robust localized estimates because of the constraining happening across sites and also between the two different types of signals. And so this was sort of where we landed as a final version of the model to be submitting to the hub, the COVID forecast hub. We did iterate on a number of different models where we kind of varied where this hierarchy came in or how we were handling it, but this is where we sort of landed back in the late fall, early spring of this year, or early winter. Okay, and then I kind of glossed over this, but I just wanted to bring it up that I sort of glossed over the whole, like, from actual shedding to the reported concentrations aspect. So I put it in that previous slide as just being modeled as a deterministic transformation from incident infections, where in reality, there's a ton of different factors that are going on between virus being shed into the wastewater and then that being reported. And so here's just showing like an example of the sort of pipeline that we believe that the wastewater samples go through. So there's some component of it that is how much time does that sample spend in the sewage network? How is it collected? There's different collection methods. There's grab bag versus 24-hour composite. How is that extracted? What targets are used? How do we concentrate that sample and amplify it? What sorts of quantification methods are used? So, for example, what PCR type are we using? And then lastly, how is this reported and at what cadence? And so at each of these steps really is like a separate module of a wastewater data generating process. And all of these are like really things that we would like to develop on. I'd encourage everyone here to take a look at Adrian's EpiSewer because he does a lot of these and it's super cool. And I really think there's a lot of fun new science to push on this. And every time we talk to people that are generating wastewater data and analyzing it, they always bring up like, hey, wait, how did you account for these different things? And the way that we did is we kind of gloss over it. We use statistical model where we basically say that the actual observations, so the concentrations in site I processed in lab J at time T, are distributed log normally with a median given by that deterministic expected concentration, that's that C sub I, and then multiplied by a scaling factor that's specific to the site and lab combination, MIJ, and then some log standard deviation given by that sigma ij. And so for what this is really doing is just saying that we're actually saying that we think that the deviations and the absolute magnitudes of the concentrations don't reflect differences in prevalence, but they're actually just differences in this sort of wastewater measurement process. And so we're really using just the trends. And likewise, that the observation error in the observed concentrations is a function. It can be is both intrinsic to the actual population in the site, but also due to the measurement process. And so we're just going to estimate that. And so what this means is like we're sort of just saying we're going to take the trend in these concentrations and use that to inform our estimates of the RT. So I just wanted to pause there and stop and see if like anyone had questions on the actual structure of the model that we chose. Okay, hearing none. All right, Adrian, you unmuted. Go ahead.
One thing that I wrote in the chat. So you said that you model the infections also in the unobserved part of the state, right?
Yeah.
And there I was wondering, do you basically assume or like pretend that this is then just one additional large catchment that you don't observe, and like it also has like a deviation from the state level trend that you draw from the same kind of like distribution that you use for the observed catchments?
Yeah, exactly. And so that's exactly how we do it. So it's basically the population of the state is partitioned into like N sites plus one subpopulations, and all of those are centered around that state, like global RT. And so that catchment area that is not covered by wastewater, that subpopulation, is just a deviation from that state RT. In practice, like when we look at it, like that one is often relatively, it's closer to the estimate of the state RT. So the deviation tends to be smaller, I think, if the population is large, because that's the one that's giving rise to the majority of the hospital admissions. So it's most informed by the hospital admissions.
Thanks.
Yeah, and that was something we changed like midway through the production of the forecast. We were initially just treating all the subpopulations as independent. And then we realized that like the population size and their relative contributions should be accounted for. Okay, I'm going to keep going. Oh, go ahead, Sam.
Sorry, I think this might be worth clarification, but could you just touch on this site level, the site lab specification, and the advantages and disadvantages of thinking about it like that?
Yeah, sure. So I mean, I think the way that we're doing this now is we are lumping together, we have observations that are assigned to a site and a lab. And there's also a bunch of other metadata that includes stuff like what kind of PCR is being used, what the collection type is. I think I mentioned like composite versus grab bag. And what we're doing right now is just saying that within a site and lab, you're going to have one site lab level multiplier and one site lab level observation error. And in an ideal world, we would actually, because these lab methods can change within a lab, we would actually have components of the model that accounted for the impact of, say, switching from qPCR to digital droplet PCR. Right now, if that happens, our model is not able to like transition because this is not time varying, and we're not incorporating that metadata. So there's definitely some limitations here. The reason that we even thought, oh, wait, we need to do this is because the CDC's contract switched from Biobot to Verily. So a lot of these sites have this discontinuous data stream where they're like being estimated or being processed at Biobot, and then they switch to being processed at Verily. And because that is an entirely different set of processes, like the actual magnitudes are not comparable. And there's lots of work in the wastewater field to try and make them comparable when these things switch and do normalizing and all of those things. And we just decided to make it as simple as possible and not and handle that statistically rather than try to either manipulate the data, which never feels like a good idea, or to model those things explicitly. Okay, I'll keep going. So next, I'm just going to like describe a little bit about what our workflow was. So this is the same plot I showed before. I'm just highlighting that, woohoo, like we're in here somewhere. So we started submitting these to the COVID forecast hub in February. So what we did in our sort of regular weekly pipeline was that each week we fit both the hospital admissions component of the model. So we removed the wastewater component of the model, as well as fitting the full wastewater model when wastewater data was available. And then we would meet every Monday morning and we would look at the convergence and diagnostics of the wastewater model in each of the states and identify issues that we had, as well as look at the forecast comparing both the forecast from the hospital admissions only model and the one that's jointly fit from the wastewater and hospital admissions. And we would discuss which one we would want to submit. Although we tried to, in most cases, submit the wastewater model, we did sometimes either find that one, the model wouldn't converge, two, the model, the wastewater data looked like there was some issue in it that we didn't believe to be due to infections, which is like a whole nother, I think, area of science to sort of explore, or some other reasons. So we basically use that hospital admissions model as like a backup to submit so that we could submit for all 50 states, DC and Puerto Rico each week, but also as sort of like a baseline comparison in real time to sort of provide us with like a stringency test of like how are we doing in comparison to this other relatively similar but pared down model. So we did this from February till the end of April, submitting these forecasts each week. We stopped at the end of April because the NHSN stopped requiring hospitals to report hospital admissions, and so the hub stopped accepting submissions. And so we've been focused since then on really, because we generated forecasts of an observation, that we can really use this to evaluate the underpinnings of the model. So evaluating what the added value of this wastewater was. So that's really like what our key question in this evaluation is how does wastewater, and specifically the wastewater model incorporated into our model, impact the forecast performance of hospital admissions. So what we've done is we've retrospectively generated forecasts from both the model with only hospital admissions and the one jointly fit to wastewater and hospital admissions, using vintage data sets for both the hospital admissions and the wastewater every Monday from the beginning of October to the end of March, or to the middle of March, where we have complete evaluation data. And we did this for all states and dates that had wastewater data. So we're trying to condition on the presence of wastewater data. And then we scored the log transformed posterior samples coming out of this model for each day of the 10-day nowcast period and the 28-day ahead forecast period. And then we're stratifying these by horizon, forecast date, and state, and starting to take a look at those results. So I just wanted to give an example just to sort of describe this process. So here's just showing outputs from a single forecast date. This is from a forecast date in the middle of January from 3 different states. The black dots indicate the data that we had as of the forecast date, and the open circles indicate the data that we later observed. And so the first one, the top one, indicates this is showing where wastewater data actually can be a leading indicator in the change in the trend. So it seems like this downturn in this particular site, and I'm only showing 3 here, but this is just a snapshot. There are other sites in each of these states, was able to detect a change in the trend in concentrations, which leads to a forecasted downturn in the admissions that the hospital admissions only model wasn't able to pick up on quite as obviously. Whereas in Virginia, we see that the wastewater data really isn't providing much information here. And so the 2 forecasts look really similar. And then in this example in Washington, we actually see that the wastewater data is misleading the forecast. So here the wastewater data is still increasing, all the data available up to that time. And therefore this leads to an overprojection of the model, projecting that the hospital admissions would continue to increase, whereas what we really saw was a stabilization or a downturn in the admissions. And this is sort of a theme of the analysis, is that sometimes the wastewater data has the ability to improve the forecast, and sometimes it does nothing, and sometimes it misleads it. So here are like preliminary results from doing that analysis and scoring every single day for each forecast date and state for both models, and then computing a relative CRPS score. And as you can see, this is the overall results. So this is the distribution across all of those states, forecast dates and days, stratified by horizon here, and then this one overall. We see that on average, we're doing about the same, sometimes better, sometimes worse. And that's kind of clear when we stratify across the states. And the one thing that I'll note here is that we do seem to see somewhat of an improvement in the month of January across states and across days and horizons in the wastewater model. So when the relative CRPS does dip below 1 here, and this is something that we're like looking into and had hypothesized that perhaps the wastewater model could improve the estimate of when, say, a peak is going to occur and the trend will change. But this is all like still preliminary. We're still sort of parsing through how we're doing this analysis, but these are sort of the preliminary results. And then the next question that we had is, how did this model compare to the other hub models? So we have the ability to do a real-time comparison because we have the submissions from February to March, but of course we can't compare those to the submissions from the entire season. So we did a stratified comparison for just February to March, where we included our real-time submission and also retrospective submissions for just that period. And then we also generated the forecast from October to March, plus what we would have, and compared them to the hub models. And this analysis is just a little bit different because in the last one, we condition on the presence of the wastewater data. But in this one, we are saying, equipped with the wastewater model, what would we have submitted? Go ahead, Sam.
Just flagging it's 3:40. So
okay.
Yeah, if you wrap up in the next like 5 minutes, so we'll still have 15 minutes.
Yeah.
Or keep talking.
I'm going to breeze through this. Basically, these are some of our results we looked at. The dark green line is our retrospective wastewater model. And the pink is the UMass model, which is one of the better models in the hub. And then the light blue is the 4-week COVID hub's 4-week ensemble. And then this light green was our actual real-time submission. So from February to March. And we sort of are looking at like, when does the model perform better? Again, this is sort of demonstrating what we think is a consistent trend, which is that it tends to perform slightly better during the peak. So this is around January, as I mentioned, but we are still sort of looking into this. So the goal here would just be to see like, okay, how do these models compare to other models? What we're seeing so far is that the hospital admissions only model, not surprisingly, is a pretty good model in it of itself. And so that's that orange line here. and also over here. And so they're relatively similar, which is consistent with what we find in that head-to-head comparison. Okay, I'm gonna slow down a little bit and sort of talk about the like bigger picture part here, which is what did we learn from all of this? So yeah, these are basically like mistakes or things that we would do differently. So we started with a code base that was initially, it was literally called COVID wastewater prototyping. We were just playing around with like models, and this turned into our production level pipelining. And so the model development was embedded with the pipelining code, and this made things really messy and very brittle because it was hard coded in to be a very specific in a very specific format that I had written when I was trying to just see if I could get these models to fit. And we put this out there. We made the code base public when we produced the forecasts, although we weren't doing really open development then, but we did make a vignette and we showed basically how one could fit their own data. But I think that this proved somewhat difficult and time consuming just from conversations with those that did try to do this because they had to finagle their data into like exactly the format that we had it in. And ideally, these two things should be separate. So the way that CFA has a pipeline for EpiNow2 to produce RT estimates, but EpiNow2 is separately developed and worked on in a different code base. I think that would have helped us a lot. And so going back, we're trying to now separate these components and develop a modeling package that just does that. And then also we found that modeling and developing on the model, improving on the science was really hard during production, especially because we didn't have any performance benchmarking. So we were not, every time we made a change, we were not able to just rerun all of our previous forecasts using vintage data and compare what our performance would have been. And this is where I think we would have felt more confident as we were trying to make changes, because we would have known that it was at least the same or better than what we had currently implemented based on some actual like quantitative evaluation. Whereas a lot of what we did when we did make small changes were just based on vibes and looking at the outputs and comparing some outputs from some states, from some forecast states, and being like, oh, it seems to do better. And also we think it is more aligned with our idea of reality. And so I'm gonna like breeze through this, but basically what we're working on now is where I think this piece is really challenging is like, we have put this out there, we've gotten a lot of feedback, and we've gotten a lot of requests for new features. Some of these can be like kind of parsed into only a few archetypes, but there's interest in forecasting hospital admissions at more granular levels. So if certain states have county level data streams, for example, in the US, and also interest in integrating different data streams. And then Christian's been working on integrating spatial correlation structures using spatial data, because some places actually have data on where the wastewater sites are located and believe this to be useful and informative, of course. And we're also trying to extend this to flu, which as written, it should be extendable, but in practice, flu has 2 different PCR targets. And so we're going to have to write some modifications to the model to incorporate multiple strains. And then generally just flagging that at CFA, there's sort of 2 projects going on to sort of tackle this question of how do we integrate multiple signals and then also develop modeling tools that are really modular so that for each of these specific feature requests and use cases, the model can be easily modified. And this is a point of like a little bit of not a pain point, but a place that I think are challenged by is we've made something to solve a specific problem, but now we want to generalize it because we want other people to use it and they might not have this exact same specific problem. So I'm just going to stop there and like go back to the discussion questions and leave them up. yeah, also just thank you to everyone that's been involved in this project. It's been super fun, and I already introduced everyone, so I don't need to go through and do that again. But this has been a lovely team science effort.
Awesome. I think there are quite a few questions in the chat, Kaitlyn. If you wanna look through there, there was a good one from Thomas about subsampling sites.
Okay, I'm going to read it just so that I'm not sitting here in silence. Have you experimented with Sentinel subset of sewer sheds for this process and impact on model performance? We've had wastewater sites cut in the US, so the question always comes up, how many and which sites should we keep if we can only fund N sites? We had that exact question. So we actually tried to do this analysis. We did do an analysis like this for our colleagues at News. They were interested in the impact of a reduced wastewater network. And so what we did was work with them to get actually like scenarios where they cut down the number of sites. They decided them based on criteria that they like separately developed into scenarios. So for example, like maximizing the population coverage, maximizing the geographic representation across the US, maximizing using like the Center of Excellences for News. And then we took those subsampled sites and then ran the model and evaluated the forecast performance under the different scenarios. And I will say we did find for this particular model, there were some instances where having fewer sites actually performed better. So we sometimes had convergence issues with a large number of sites, which is some science that we still need to work on. And so that would sometimes improve it. But I think that general question is one that we get a lot, and we'd like to be able to provide tooling so that people could answer that question based on their own scenarios of which sites would be cut out and what's the impact.
Adrian, do you want to ask your question?
Can I ask a different question, which I find more interesting, just because of time? What you touched upon regarding this evaluation during the model development. Yeah, I think would be just interested to hear more your thoughts on this. So it seems to me that to be able to do the model evaluation, you already have to go quite a long way of building up the models so that you can get all the way to producing, say, forecasts. So yeah, I think that was one of my questions, whether you experience this as a limitation or as a challenge. And then the other thing of how much quantitative performance scores, like in forecasting, actually helped you in maybe identifying limitations or problems during the modeling, like how useful it was really like for the actual informing the model development in the end?
Yeah, I want to say, I think that's always the goal. I think the goal is if you can evaluate, then you can more easily and more like rigorously and quantitatively improve. In practice, I think during production, and even now, I do think that we still struggle with the speed at which this process happens. So that iterative, make a change, evaluate, and then assess whether you want to merge that change is not super streamlined for us yet. But I do think in theory it should be. And in theory, every single scientific question that we have that we want to develop components of the model for, we should be putting into some type of benchmarking where we can see how it's impacting the performance. I know that Sam might have thoughts on this. I know this is something that they have set up in GitHub Actions in EpiAware. And I think that's like the dream, is not have it be this like really like exhaustive analysis that has to happen every time, but something that is automated that we've set up and we've run on some subset of the, like in this case, there's a lot of data, it's a lot of models we had to rerun to do this evaluation. And so it's not necessarily feasible to do that multiple times, but I think there's a compromise where we subsample the data and we do some type of benchmarking against it. Sam, I don't know if you have additional thoughts there, just because I know you've thought about this.
My only thought is that it's important to be clear that a benchmark is not the same as a unit test. So you might have like a test where you check it works or not. But I think an additional really useful functionality is where you run the same on the same compute, you run the same thing on both your kind of like main and feature, and then you test that. So it's like not quite the same as unit testing, but I found it really helpful for model development. I put some stuff in the chat about specific tools.
Thanks. John, do you want to ask your question?
Great. Thanks very much for the talk. That was really interesting, and it was nice seeing some of the challenges you faced. I think we've had some similar ones, and it's nice to know we're not alone. I was wondering, it's quite a specific question on that decision you make when you have your wastewater informed model and the not wastewater informed model, and you have to make a choice on what to submit. Could you elaborate a bit more on how you make that decision and what the process is? Because we don't have one, is why I ask. It's a guess.
Yeah, I mean, we wrote up I tried to write up a SOP to sort of somewhat make this an algorithm. So the first check was we had convergence thresholds for the MCMC diagnostics. And if any of the wastewater models in a particular state did not pass those thresholds, we would flag them, and we'd actually open up the diagnostics on each parameter and take a look and see, okay, what do we think is going on here? Oh, it looks like the model is struggling at estimating the RT in this site because it has this crazy behavior. So we probably shouldn't submit it because there's something going on there. But I think we also did a little bit of human judgment. And I think that did come in where we would look at trends in the hospital admissions and maybe look at the trends in wastewater and be like, okay, we actually think that the hospital admissions model is more reliable and therefore we should submit that. And I'm not sure if this is the best way to do things. I think I'm sort of hopeful that maybe next season we would submit both models and actually do a real-time comparison. But yeah, I think we could have streamlined that decision-making process a bit more. We decided we'd all discuss, and then Dylan would make the final decision. We needed some sort of there's a sense of authority. Someone has to decide what we're actually doing. But it was a lot of discussion involved and looking at these outputs and looking at the diagnostics.
Great, thanks.
Go ahead, Sam.
Yeah, I have a question I'd like to ask your opinion. So we have this package at EpiNow2 that was written a while ago, and then that's receiving continuous development. I also have a package called EpiNowcast, which does broadly the same thing, that's receiving continuous development. Adrian has a package called EpiSur, which does broadly the same thing to both of those, but for wastewater data, with a lot of complex stuff going on. And then you have this package for wastewater data, which does broadly the same thing as EpiSur. It doesn't have a hex logo, so it's bad, but has some other stuff. When is it? And then there are lots of other people have lots of other tools, but all of those tools are in Stan. All of them share quite similar ideas. None of them have any overlapping code in the sense of they don't have shared dependencies.
Yeah.
When is it that we should be thinking about how to better integrate? And I think in particular, what I'm interested in is when you begin a project like this, it's always easier to kind of hack it And when you want to put in a new feature, it's easier to just put that feature in and not worry too much about like the broader ecosystem. But if you keep doing that, eventually you find yourself with these independent forks, all of which have lots of complicated functionality and none of which is interoperable. Yeah. So how do you, one, how do you avoid that? And two, do you even want to avoid that? Does it matter?
Well, okay. First off, I think that there is some value sometimes to prototyping something first to show that it can work. And sometimes I think it's okay to do the easier thing, which is roll your own. Shockingly, like it is always, I think, easier to like for this project instead of branching off of EpiNow 2. I just grabbed all the things that I wanted and stole them and developed off of that. And I think it allowed there to be something that this thing could produce that we could discuss with news and we could discuss with our leadership and we could say, we think this is worth like investing in. I think the question of when do you pivot to trying to develop interoperable, modular, generalizable software is like the thing that I still don't know the answer to. Like, I mean, I don't know the answer to many things, but like, that's one of the questions that I wanted to ask this group is like, yeah, maybe there should have been a point where, after we developed the single population, like single wastewater data stream model that we decided, okay, let's see if we can make this integrated into the existing landscape of epi modeling tooling. And I do think that there's probably better ways to do that than what we're currently doing, which is writing all of our own code. So perhaps that's Julia. Perhaps that's like some type of shared infrastructure. I'm not sure. I don't know if I know the answer to that question, but I think it's a really good one because it's also related to what is your long-term goal, right? So if you want this to be something that people could continue to build upon and then like it is the more general thing, then I think it's worth having that conversation a lot earlier. But if your initial goal in the short to medium term is like doing something for a specific thing, then where do you find yourself? Is it easier to just develop on your own, or should we be pushing more towards this interoperability? And I think the answer is yes, we should be. But in practice, it's hard.
Yeah, it makes sense. And as a follow-up, how do you think you should push towards interoperability? Like, what are the steps? Like, everyone says, like, yeah, it's a good idea, cool, at conferences, and then we go home and we like write our own software packages. So,
Yeah.
or like methods and papers, and then we say they're the best. So, what is it that we can do? I don't know if anyone else has ideas. I think it's quite a hard question.
Yeah, I mean, I think like this is helpful, like having shared communities where you, at least if you're building your own software packages, are not starting from scratch because you can find where someone did it well and you can, I mean, Sam knows this and Zach knows this, like my go-to thing is just copying things that already exist that I like. So I think like knowing about that and there being a community for this is one step, but that's not interoperability. That's just like shared knowledge bases and shared coding. And I think we even struggle with this within institutions. Like it's not just a problem across groups. So I don't know. I don't know if I have a good answer to that, but would definitely throw it back to the rest of this group.
I might have one more thought that there's also like the difference between actually building the statistical models and then building interfaces to them. I mean, so your comment on all these models, I understand. I mean, mostly goes this direction of, okay, how can we actually merge the components of the statistical models? But I think in terms of development time that's being invested, it's also really the interfaces. Like, for example, if you nowcast, just a lot of open issues and work are really on that are like related to the usability, are really related to just building an interface in R around it. So maybe in terms of interoperability, being able to, maybe even if we can't merge different statistical models that easily, being able to access them in a joint interface so that not everyone is just building their whole package around it could already save a lot of work. And then also increase the comparability and all this other stuff.
Yeah, I mean, even if outputs were standardized, and I mean, in not standardized necessarily, but in some way you could, and this is, we've been talking about this with RT estimates, like, can you find a way to develop a single data set that can be used across all the RT estimation packages, and then you can actually compare them and deploy them easily. Like, say you have a production pipeline, you could line up and do all these models and be able to, yeah, I think interface with all of them, even if under the hood the underlying models have different code bases, and that might be sort of on us to try and figure out how we should be outputting things to make them maximally interoperable, even at the level of, like, not the functions or the objects, but the output. I don't know. Easier said than done. James, I'm not sure what tidy models is. Do you want to describe it? I know we're over time, but.
It's just a thought, but I think it's a move towards having a more standardized frequentist interface. And not really frequentist. I think there's a lot of machine learning stuff in there as well, but the ability to specify stuff and pass them around between packages without having to do a lot of wrangling. So there's like a standard for doing that people have to follow. But I like Sam's idea of us thinking about the common tasks that are often performed across packages, like specifying priors, for example, or delays and stuff like that. And in Epinal 2, there's a move towards having a standalone distribution interface that you can specify distributions through that you can pass to Stan. So yeah, there's a lot of work to be done in that regard, but it's doable. It's just, I think it has a lot to do with ownership and credits, which has been a conversation in the past about how do people get credits. So for example, in Epinalcast, there's the move towards community credit and people getting credit on the community level instead of individual level, but people have different motivations for doing these things and different performance metrics. And if that is not properly sort of discussed and set up, it's going to either discourage or encourage people from contributing to these community efforts. And I think one thing as well is about control. So if we remit a lot of these workflow stuff to other people that are developing the packages, you have to depend on those packages to fix things upstream before you can use them. But quite often, if you have it local to your organization, then you can do it in-house. So there's that aspect as well, which, yeah, but I mean, there's a lot of things to discuss in this area. I've been thinking about that quite a lot lately.
We are well over time, but I just wanted to say an example of a really boring thing here that I think is a really useful action is all of these tools need to install commands down into CI. That's really boring, and they need to cache it so that they run a bit faster when they do their checks. I think that is like having an action in the Epinacos community that is like maintained is like a bit of a labor saver. And I think there are quite a few things like that that you could do that you could share across packages to save people time, even if it's not directly related to the methodology or the model, but they're related to kind of the infrastructure you need in order to have a good model, as long as they will share the same kind of like backends. Right. Does anyone else have any more questions for Kaitlyn, or shall I stop recording and wrap up with a emoji round of applause?